So this was a phase 1 clinical trial looking at the use of autologous-derived CD5 CAR-T cells for the use of relapsed or refractory T-cell ALL. In the initial cohort, we manufactured the CAR-T cells using standard manufacturing processes with IL-15 and IL-7 stimulation. While the CAR-T cells were well tolerated, we saw minimal responses with only one MRD positive remission achieved. In subsequent analyses, we discovered that the CAR-T cells were undergoing chronic signaling, which was leading to an exhausted T-cell phenotype, which ultimately resulted in impaired efficacy. Following that discovery, we looked at the use of adding TKI inhibitors, particularly dasatinib and ibrutinib, during the expansion process in order to inhibit the upstream T-cell receptor signaling kinases responsible for activation. And after we made that change, we saw a significant improvement in the phenotype of cells, being a more naive and cytotoxic phenotype. And we also noted that while there was no significant difference in the expansion seen, there was a significant improvement in the persistence of the CD5 CAR-T cells. And so in the subsequent cohort, we treated 7 patients with TKI manufacturing, 7 from autologous PBMCs, and we actually had another cohort of donor-derived CAR-T cells, all using TKI manufacturing though. And once we made that change, we saw an improvement in the response rates to 57%. 4 of 7 patients achieved an MRD negative remission compared to only 12.5% before.